Stanford University.
Well, nice to see you all here again. I heard, actually, while I was in Asia last week, that the presentation by Mike Snyder went very well, so I'm glad to hear that. And today we're going to move from the host to actually a population that is more numerous on each of us and in each of us than our own host cells. I mentioned in passing at the very beginning of this course that actually microbes are more numerous on our bodies than actually our human cells. And so this is an opportunity for us to begin to delve down into this companion that lives with you and on you and through you for all of your life, does lots of good things, and then also creates a little bit of havoc. Obviously, we're all pretty focused on a virus these days, H1N1, and we could talk about that perhaps later, but there are many wonderful things that happen by way of microbes. There's an interesting book that I recently read by Stephen Johnson, who writes some sort of popular science, and this one was called The Ghost Map. And I'd recommend it to you, particularly after Julie's presentation. It's the story of cholera around the 1850s in London, and it's as much a depiction about microbial host interactions as you're likely to witness in a very thoughtful and somewhat provocative way. And one of the interesting points that he makes in passing, and I don't know whether Julie will agree with this or not, is that if you were to, in essence, wipe out all the mammals in the world today, it wouldn't really change the world's biodiversity. But if you eliminated the bacteria, you'd wipe out the world in pretty short order. So the bacteria, which are the oldest of our evolutionary species, are really critically important and the topic of Julie Theriot's work. Now, Julie, who's going to be our speaker tonight, comes to this by way of an interesting background. She actually co-concentrated, received 2 degrees from MIT, one in physics and the second in biology. And then kind of fused that as she moved to her graduate work at UCSF, where she concentrated on cell biology and really became more interested in microbes, which has really been the story of her own research career. And she has not forgotten her roots because she's really combined physics and computation and biology and lots of other tricks and trades along the way to become really one of the world's leaders in this field. She's currently an associate professor in biochemistry and microbiology and immunology, but is also the recipient of some enormously distinguished awards, even at this early stage of her career development, including a MacArthur Foundation Award. And you know, that's meant to be just for that small population of geniuses, and I think she certainly achieves that title. She's also been elected to be a member of the Howard Hughes Medical Institute, so another distinguished accomplishment in what I know is going to be a continuing legacy of great achievements. She's highly recognized as a teacher to our medical and graduate students, and I'm very pleased to have Julie speak with us tonight. So.
Well, thank you, Phil, for that very nice introduction. And I'm very happy to have the opportunity today to come here and talk to you about the microbes that live inside of all of your bodies. Now, I came to a couple of earlier incarnations of this in previous weeks, and I noticed that this is a very interactive group, and you're very curious, and you like to ask questions in the middle. I want to strongly encourage you to do that. I can't tell you how happy it makes me to get questions when I'm in the middle of talking, because that way I know that you're actually listening to me. So please don't feel at all shy about stopping me and asking questions in the middle. Now, the task that I was given today, and in fact the title that I was given to discuss with you, is this concept of the world within us, specifically to talk about the microorganisms that live as part of our bodies and are critical both to our normal functioning, our normal physiology, and our normal health, as well as causing disease. Now, I'd like to start off just with a naturalist's observation. This was made by Jonathan Swift in 1733. He said, "So, naturalists observe a flea has smaller fleas that on him prey, and these have smaller still to bite him, and so proceed ad infinitum." This image, of course, is an electron micrograph of a flea, which is a common parasite of mammals, including humans, dogs, rats, many other mammals. And it's also one of the organisms that's responsible for spreading some kinds of human diseases in populations, most notoriously for spreading the bubonic plague. Normally, rats are covered with fleas, but when the fleas started giving the plague to the rats would die, and then the fleas would hop off the rats onto the nearest convenient mammal, which in cities would tend to be a human, and thereby spread the plague through cities. Now, my distinguished colleague Stanley Falco, who's an emeritus professor in the Department of Microbiology and Immunology, early in his career was studying bubonic plague, and in order to study the way that the bacterium that causes bubonic plague, that's Yersinia pestis, actually goes from one mammal to another, it was also necessary that he study how it survived inside the fleas that carry it around. So Stanley very proudly cultured a colony of fleas, and he was very fond of his fleas. He would feed his fleas carefully every day and take very good care of them. And then he became very sad when he noticed that his fleas seemed to be sick. So he took his fleas, although he was very fond of them, he had no qualms about killing them, and put them under the electron microscope to see what was going wrong with his pet fleas. And this is the picture that Stanley took at the time. This is now the leg of the flea, and you'll notice it has smaller fleas that on it prey. These are mites, and it was a mite infection of his flea colony that was causing the fleas to become sick. Now that's remarkable enough, but one of the things that I'm particularly fond of in this photograph is that, at least those of you in the front row, if you look very carefully at the surface of the mites, you'll see these little specks just right on the surface. Can you see those? Yeah, so those are bacteria, and those are bacteria that are living on the mites. Now my group, as Phil mentioned, studies bacteria. And if you look very closely at bacteria, so here now these are a couple of E. coli cells. This is the outline of the bacterial cell, and these long extensions coming out of it are flagella that enable the bacteria to swim around. But if you look very closely at bacterial cells, you'll often see that they too have little parasites. In this particular case, this was an experiment that was done by one of my graduate students where she deliberately added these little specks you might just be able to see in the micrograph to the bacteria actually to infect them. These are bacteriophage, which are a particular kind of virus that only infects bacterial cells. And here this is a much higher resolution electron micrograph where one of these little creatures here that looks like the lunar landing module corresponds to one of these little specks here. So we've gone from the flea to the mite to the bacterium to the bacteriophage all the way down as Jonathan Swift predicted. So I'd particularly just like to emphasize that when you think about any particular organism on Earth, for example, when you think about yourself and your own body, it is the natural state of affairs that you'll be covered with smaller and smaller fleas, and so proceed ad infinitum. Now, in particular, in the case of the human body, as Phil mentioned, we tend to like to think of ourselves as being individual organisms. And I know when Mike Snyder talked with you last week, he encouraged you to think of yourself as a single coherent organism that has one genome, where that genome is replicated in all the cells of your body. Well, Mike was right about 10%. But only 10% of the cells in your body actually have human DNA in them, because the other 90% of the cells in your body aren't human at all. They're bacterial cells, they're fungal cells, or they're protozoal cells. Now, the exact numbers, you'll see different kinds of estimates. The number of human cells in the human body are thought to be about 10 to the 13th to 10 to the 14th. The number of bacterial cells, primarily with a little smattering of these other kinds, is about 10-fold more. So rather than thinking of yourself as being an individual organism, it's actually much more appropriate to think of yourself as being a complex and thriving ecosystem, of which the human cells are only a minority. Question?
How much is it by weight of bacteria?
Okay, that's an excellent question. So the bacterial cells are very, very small. And typically, an average bacterial cell that inhabits your body is going to be between 1/100th and 1/1000th of the average weight of one of your human cells. So overall, it's somewhere on the order of a few % of your weight is made up of bacteria. But if they were a democracy, if every cell got an equal vote, then the bacteria would win every time. Now, does anybody have any idea where the highest concentration of bacteria in your body is to be found? Yeah, so when you produce feces, actually a big old chunk of that is bacteria that are coming out of your lower intestine, which is where the highest concentration is. And there, you probably can estimate what volume or weight of feces you produce every day. You'd say probably about 30% of that is bacterial cells, and you can do the calculation yourself then to figure out how much of your body weight that represents. So in this context, I'm particularly moved by this quote from Walt Whitman's Song of Myself, "I am large, I contain multitudes." So as you walk around, just remember you're never alone. You always take your whole ecosystem with you. Now, most of the people in this room, I assume, even though you're completely covered with bacteria, they're all over your skin, all over your mouth, all through your digestive tract, you're probably reasonably healthy. And actually, to a first approximation, having those bacteria as co-inhabitants with your body is important, is necessary for maintaining health. And as we'll see, also for performing certain functions, particularly in the digestion of food, that you're not really that good at doing with just your own human DNA. So what I'd like to talk about today is going to be divided into 3 parts. In the first part, I'm going to try to introduce you to the microorganisms that live on your body. Now, you're, of course, intimately familiar with them in one sense, but probably don't know that much about who they are and what they're doing there. So I'll start off with that. And then, whereas we're going to start off focusing on the benign microorganisms that we live with all the time, obviously an interesting and important aspect of bacteriology as far as medicine is concerned is those few bad actors, the rare bacteria in the world that cause disease in humans. So I'd like to discuss a little bit about our current understanding of how disease-causing bacteria or pathogenic bacteria are different from all of these many, many trillions of benign bacteria that cover us all the time. And then finally, I'll move on to talk a little bit about how human cells and how the human organism interacts with both the helpful and the harmful microorganisms. And this will be a little bit of a segue into the talks that are coming over the next couple of weeks from Dr. Gary Skulnick and Dr. Lucy Tompkins, who will give you different kinds of views of the interaction between humans and microbes. Now, I'm a cell biologist. My training is in cell biology. So I tend to think of humans, and indeed all organisms on the planet, as just being big piles of cells. And here today I'm representing the bacteria, if you will. So I'd like to start off by putting bacteria just sort of on an equal footing with all other organisms and all other cells. So humans, remember, it's about 10 to the 13th cells or so. And as you know, those cells are quite specialized to perform different functions. But if you look very, very closely at any particular part of a human being, you can easily see the granular structure that makes us up. So this, for example, is a thin section through a human kidney, where each one of these open white circles is an individual nephron, which is where the urine is formed and is filtered. And then around the outside of each one of these things, you see there are these little squares that have a circle in the middle. These are kidney epithelial cells that are involved in absorbing and secreting salt and water to form the urine. So each one of these little round spots is a nucleus, and that has an entire complement of the human genome in it. And you see the cells cooperate together to make these nice rims around the kidney tubules. And if you were to look in any of your favorite tissues in the body, you'd see a similar sort of organization where lots of cells are working together. Now, the other major prominent multicellular organisms on this planet, besides animals, are plants. And plants also are basically a cellular structure. In fact, it was originally Robert Hooke describing the tissue of plants, the structure he saw under the microscope, where he coined the word cell. And this is just to show you the cells of a plant. This happens to be from a leaf, where you can see the outline of the individual cell here. And all of these little red bodies inside the cell are chloroplasts, which the plant is using to do photosynthesis. But again, it's just a big old pile of cells. Now, because we ourselves are macroscopic organisms and our eyes are able to only see things that by absolute standards really are quite large, we tend to think of the planet as being inhabited largely by big things, by animals and plants and maybe the occasional fungus. But in reality, as Phil alluded to, almost all of the living creatures on this planet are in fact microscopic, and most of them are unicellular. Now, many of those unicellular organisms are eukaryotic cells, like us. That is, they have their DNA carried in a nucleus. We'll talk a little bit more on the next slide about what specifically is meant by eukaryotic cells. But I'm just giving you some examples here of what some of our relatives look like who are not multicellular. This is Tetrahymena, also known as pond scum. Any of you who were in the habit of taking samples out of the creek in your backyard and looking at them as a little kid saw lots and lots of Tetrahymena swimming around. This is Saccharomyces cerevisiae, or baker's yeast, or brewer's yeast, which is very good at converting sugar into alcohol for our own pleasure. This Emilion huxleyi is a very interesting unicellular organism that's a coxolithosphere, which causes algal blooms out in the ocean. And these things can be spectacularly large. You can see them on satellites from space. There was a recent algal bloom in the North Atlantic that was actually the size of the state of Alaska. And it was just made up of many, many trillions or quadrillions of copies of this little individual cell. So those are all eukaryotic cells. This up here is Escherichia coli, who is a bacterium that is much beloved of laboratory scientists. And we'll talk about that actually quite a bit more. But this is a bacterial cell on the same footing as all of the other cells. So I'd like to just briefly mention the major distinction in cell biology based on cellular morphology between prokaryotic cells and eukaryotic cells. So we, other animals like whales, including invertebrates like the fruit flies, and then also plants like this redwood tree, we are eukaryotic organisms, which means we have all of our DNA enclosed within a membrane-bound compartment, or the nucleus. Now that is a choice that our ancestors made on the order of between 2 and 2.5 billion years ago, but it's not a particularly important choice, and it's certainly not the only way to be. All of the rest of the cells on the planet are prokaryotic, which means before the nucleus. And these guys have their DNA just in the cytoplasm, along with the ribosomes and all of the other metabolic enzymes and all the functioning parts of the cell. Prokaryotic is a commonly used term, but it's actually kind of an old term. And we appreciate now that the wide variety of prokaryotic cells can really be better described as being divided into 2 large kingdoms. And those are the bacteria and the archaea. And I'm going to focus today mostly on the bacteria because those are the ones we understand best, but I will say a few words about the archaea here and there. Now, the easiest way to tell the difference between a prokaryotic cell and a eukaryotic cell, if you're looking down the microscope, is simply size. Prokaryotic cells, like bacteria, tend to be fairly small, and they tend to be fairly simple. They'll have shapes like rod shapes or spherical shapes or sometimes little spirals. But if you look down the microscope like at pond scum and you see something that's got, big long whips or is swimming around or is eating something else or is sort of changing shape and oozing, that large thing that's morphologically complex is going to be a eukaryote. So although they're small and they're morphologically simple, they greatly outnumber us. In our human body, it's probably about 10 to 1 prokaryotic cells to eukaryotic cells. On the planet, it's very difficult to actually take a good census of the total number of cells, but it's estimated to be between 1,000 to 1 and 1,000,000 to 1 that prokaryotes outnumber eukaryotic cells on the planet. And as I mentioned, even among the eukaryotes, most organisms are microscopic. Now, although these cells are very different in terms of their appearance, there are a lot of things that they have in common. And in particular, I know Gil Chu introduced you to the central dogma of molecular biology, that is the concept that DNA is replicated, DNA is transcribed into RNA, RNA is then translated into protein. That fundamental 3-step mechanism for the expression of genetic material is shared by all cells on this planet, all the bacteria, all the archaea, all the eukaryotes as well. And one thing that's actually very convenient about that, if you're interested in surveying the identity and the location of cells, for example, of bacterial cells, is the fact that because all cells on the planet work fundamentally the same way as far as the central dogma goes, that means you can count on every cell having certain components in common. And the one that's actually turned out to be the most useful for the kinds of studies that I'm going to be describing is the fact that all cells have ribosomes. So ribosomes, remember, are the big machines that take the messenger RNA and then translate the triplet code in the messenger RNA into a protein, and that protein can then go on to have biological function. And the ribosome itself is an incredibly elegant, complex, beautiful piece of biological machinery. It's extremely large. It's 25 nanometers across, which makes it about 50 times bigger than an average-sized protein. And it's not a single protein. Instead, it's a complex that's made up of many, many components or substituents. And of those components, a lot of them are in fact proteins. So for example, the bacterial ribosome that does the work of translation in a bacterial cell contains 55 proteins, but it also contains 3 large RNA molecules. Now these are specialized RNA molecules that never get translated into a protein. Instead, their sole function is to make the structure of the ribosome that does the translating of other RNAs. If we look in eukaryotic cells, they also have ribosomes, but their ribosomes are a little bit different. They tend to have more proteins, they're a little bit fancier, and they also have 4 RNAs instead of 3. But you can pretty much line up the RNAs from all the eukaryotic cells with the RNAs from bacterial cells and see how they're related to one another. Because of this, because of the fact that every single cell on the planet of every possible different kind has ribosomes in it, and specifically has ribosomal RNA in it, if you want to make some kind of measurement or some kind of calculation about how closely related any 2 cells are, one very convenient way of doing this is just taking the sequence of the ribosomal RNA and comparing that one to another. So for example, if you were to take human ribosomal RNA and compare it to chimpanzee ribosomal RNA, it would probably be almost identical. If you were to compare either of those to the ribosomal RNA from a dog, you might see a few changes. If you were to compare either of those to the ribosomal RNA of a fruit fly, now you begin to see some more changes. To a redwood tree, yet more. To a single-celled organism like Tetrahymena, the pond scum, you'd see more. And if you compared it all the way to a bacterial cell, you'd see even more changes. But the point is, they're still similar enough. You can still line them up, and you can count how many changes have taken place among all the different cells on the planet. And this is absolutely astonishing. And this has been used in a lot of very creative ways by a lot of scientists to try to understand how we can look back in time and see how evolution has occurred and see how everybody is related to everybody else. And one of the early examples of this was a really spectacular piece of work that was done by Norm Pace and his colleagues at Indiana University in the early 1990s, where they just went around taking various samples of different things, from things like hot springs in Yellowstone National Park, water out of the ocean, soil, deep ice core samples from Antarctica, and then just measuring the number of changes that had happened in ribosomal RNA from all the ribosomal RNA they found in all the cells in those samples and comparing them to everybody else. And the really profound thing that was proved by that series of experiments is that every single cell on the planet, including all of your cells and all of the bacteria and everything in between, they all are descended from a single common ancestor. That is, we can line up all of these ribosomal RNAs and we can see how they're all related to each other, and by counting the changes, we can see how they diverged. So when we're looking at this image, and this is a kind of tree that I'm going to be showing you a lot of, the length of the branches in the tree shows you how similar or how different two different sequences are. So for example, if you see something like this little branch up here, where you see these three branches that are connected with short lines, that means those three sequences are all fairly similar to one another, and therefore perhaps diverged from one another relatively recently in evolutionary time. If you look at things that are related only by very long branches, so this here, compared to this here, the longer the distance that you have to travel on the tree before you get to the other thing, the more different the sequences are from one another. So this is sort of a convenient way of classifying very large numbers of sequences to say sort of what's the pattern of diversification. Now just to orient you here, this tree from a publication from Norm Pace's lab from 1996 shows all of the three major domains of life. Where down here are the eukaryotes in this branch, here are the bacteria up in this branch, and here are the archaea over in this branch. And this kind of work was one of the things that established the fact that archaea and bacteria, although they look alike to us, are really very different from each other, and in fact have been diverging for just as long in evolutionary time as the eukaryotes have been diverging from the bacteria. So if you compare all of the cells up here, the archaea and the bacteria, again, they're wildly divergent. They've been evolving away from one another for between 3 and 4 billion years. But they look very similar, and in fact, what they've concentrated on is diversifying their metabolism. So we have extremely exotic metabolism going on in these cells. These were the cells that invented photosynthesis. In fact, there's never been a eukaryote on this planet that can do photosynthesis. And the only way that things like trees and plants are able to do photosynthesis is because a long time ago they captured a bacterium, they captured a cyanobacterium and domesticated it and had it turned into a chloroplast. But the exciting metabolism that goes on there that changes light into sugar, that was invented by a bacterium. We have bacteria that are able to eat rocks. We have bacteria that are able to generate methane. All of that complicated metabolism that's only done by prokaryotes. Eukaryotes are extremely boring as far as their metabolism goes. So what have we got going for us? Arguably, we rule the planet. I mean, here we are. We have civilization and all those kinds of things. The way that we're able to have civilization, even though we have extremely boring metabolism, is that we're very good at doing morphology. We're very good at making large cells, cells that do fancy things, cells that swim around, and particular cells that like to pile up to make big multicellular organisms. But if you look at the multicellular organisms, everything you can see with the naked eye, that basically comes down to animals, plants, and fungi. And here on this tree of all the cells on the planet, those are right over here. So this branch here, Homo, that's us, Homo sapiens, and this branch actually represents all of the animals. And I don't just mean all of the mammals, and I don't just mean all of the vertebrates, I mean all of the animals. So fruit flies are indistinguishable from humans on this image. This here, Coprinus is a mushroom. That represents all of the fungi. And this here, Zea, is corn, and that represents all of the plants. So at least as far as our ribosomal RNA is concerned, everything you can see with the naked eye is just a tiny, tiny twig off of one side of the giant tree of life. And so what I'm going to focus on here now is the bacteria. And I hope at this point I've convinced you that bacteria are interesting and worthwhile and at least should be considered as co-inhabitants of the planet along with us and co-inhabitants of our bodies. Yes?
Clarify once again, back to the LUCA, when everything was that, how did it evolve up here, up there, so it captured bacteria, how did it capture it? That's all it was.
Yeah. So the question was, how did the LUCA, the last universal common ancestor, how did that evolve into all of these different branches? So this is extremely controversial. And I don't want to give you the impression that LUCA was a single cell. It's not like there was one cell that was located at some location that then gave rise to everything that lives. LUCA was a population of cells. And I'm going to tell you now what I believe is the most likely explanation, but as I said, this is very controversial. But I believe what probably happened is that there was a period of evolutionary history where there were a lot of cells on the planet that were very enthusiastic about swapping their genetic material back and forth. They shared their genetic material among each other. So it was a population where everything was held in common, sort of communist cells, if you will. And then there was some point in evolutionary history, and again, the numbers are not clear, but it was on the order of about 3 billion years ago, maybe 3.5 billion years ago, where some cell got the really clever idea that it was just going to keep its DNA for itself, and it wasn't going to share it with anybody else. And that has been referred to by Carl Woese at University of Illinois as being the Darwinian threshold, which means that once you get to be selfish about your genetic material, then if you make a mutation that's good, then instead of giving it to everybody else so everybody benefits from it, you keep it from yourselves and then only your offspring will get it. Now once that happens, then you can begin to diversify. Because if the DNA that you make, you mutate, you adapt only goes to your offspring, then that gives you individuality and it gives your lineage some sort of separate identity from everybody else on the planet. So the sort of general consensus is that before this Darwinian threshold was passed, there was a huge amount of evolution that actually gave us DNA as the genetic material, that invented RNA polymerase, that invented protein translation and invented ribosomes. But all of this really complex evolution was all taking place under a context where you couldn't have separation of lineages, you couldn't have natural selection. And then after that happened, then you were able to diversify. Question?
That practically happened. Was everything having sex with everything else?
Well, sex, yeah, so sex is a very late invention. And it's not actually the best way to swap genetic material around. In fact, when we talk a little bit later about what the bacteria do instead of sex, you're going to be jealous because it's much, much more efficient. So yeah, so sex is only, that's only if you're diploid, it's only if you're a eukaryote, and really you shouldn't be that proud of it. So it was much more just that genetic encoding of information in the form probably of DNA and RNA was able to be freely shared. Now, there's a lot of controversy as to what that really means. So, for example, right now, every cell on Earth is surrounded by a membrane, and that membrane keeps everything on the inside. And arguably, that is how you distinguish inside versus outside and self from non-self. Now, there's very good arguments for why, even before the Darwinian threshold, there still must have been membranes around those cells. There's metabolic arguments that I can go into later, but I think it's pretty clear there were membranes. So if there were membranes and things were physically separate, why were they still able to swap things around? I don't think anybody has a good answer to that. I can't tell you how much money I would give to be able to go back 3 billion years and actually look at what was floating around on the planet, at that time. I think any scientist would do that, but we just don't know. And, the problem really is there could have been a lot of burgeoning life. I mean, there were a lot of cells that any modern biologist would recognize as being cells, but they're all gone now because once somebody made the decision to be selfish and keep the DNA, it could figure out how to do things like eat the other cells. And so all those other guys, they're gone. All we have left is the descendants of these lineages that decided to be selfish. And so at some point around then, it broke out into these 3 major branches, and then they diversified from each other. But exactly what happened at that transition, that's a very hard question.
So were these cells promiscuous or incestuous?
Oh, well, okay, so the question was, were the cells promiscuous or incestuous? I would argue that if you're a part of a collective, that there is no distinction between promiscuity and incest, so both. The question was, what role did viruses play? The origin of viruses is also very hotly debated. Again, this is something that is very controversial, but the explanation I personally find most plausible is that viruses are bits of DNA that got ambitious and decided to go out on their own. Things like transposons that then figured out a way to encapsulate themselves and then become more efficiently transmitted. So if that is true, and I can't prove to you that that's true, but if that is true, then viruses arose much later than the Darwinian threshold. So they probably didn't play any role in the initial diversification, although certainly they've been very important in more recent kinds of diversification. But you know, if you ask a bunch of virologists where viruses came from, you know, if you ask 10 virologists, you get 10 different answers. So I'm not sure that I can really answer that definitively. Question?
Basically, the LUCA, we don't know about LUCA. It's our extrapolation.
It is extrapolation. We infer the existence of LUCA.
But based on the extrapolation and based on what information we have to the nearest of the organisms, are we able to synthesize it or
yeah, so there are people who are going through all of the genome sequences that are now coming out from all of the cells that are on all of these different branches, and then trying to sort of look backwards in time and saying, you know, what are the things that LUCA must have had? Now, it could have had more stuff that's been lost. There could be things that are lost in one lineage but not in another lineage. But you can, you know, make a pretty good argument of the things that probably were there because they're present in all the lineages. And there's a website you can look at that's called COG, for Cluster of Orthologous Groups, where it enumerates all of the different protein domains that have been found in all of the branches of life. And there are things that are involved in very central fundamental processes of life. So things like the polymerases, DNA and RNA polymerase, certainly things like the ribosomal components and the tRNAs that you use to synthesize proteins, those are all found. The other kinds of things that are found are things like enzymes for glycolysis that convert sugar into energy. Those seem to be very widespread throughout all the domains of life. And there's about, I think on the COG list, there's probably on the order of about 1,000 such domains that are shared, that are present in at least some individuals of all three trees. Now, going from that to trying to recreate LUCA is fairly difficult. There are a bunch of people who are trying to do things like do a complete synthesis of DNA that they design. And there are a couple of people who have begun to have some success with that kind of thing. So you can try to make a fundamental primitive organism by building it up by saying, I think it needs this and this and this. Let's put it together and see if it works. People are trying that. That hasn't worked yet. The other way that people are doing that is they try to take individual cells that already exist and then get rid of everything that they don't absolutely need. And again, try to come to a minimal genome. And again, there's people making progress on that, but I would say it has not yet been definitively demonstrated what is sort of the minimum things necessary for life. But that's still different from saying, well, this is what the LUCA looked like, because the LUCA had its own issues. You know, I mean, it had to get energy from some kind of source. These people in the labs who are trying to make a fake minimal organism, they're like coddling it and feeding it, and they're keeping it nice and warm, and they're giving it delicious sugar and all. And you know, LUCA was like out in the wild, and so it didn't have that kind of coddling. So, you know, I don't know that we'll ever know. You know, I think we'll be able to sort of get at it by the kinds of experiments I described. But yeah, I mean, it's sort of, you want it to be like Jurassic Park, right, where you can just take all the bits of DNA and say, oh, let's recreate LUCA. But I don't think that's really even conceptually possible. Although I'd love to be proved wrong about that. Any other questions? Wow, okay, this was just an introductory slide. So we'll see, yeah, we'll see how this goes. All right, so let's, yeah, question? LUCA. Oh, LUCA just stands for Last Universal Common Ancestor. Okay. Okay, so here we are back to the humans, and I'm particularly going to focus on the human digestive system, in part because that's where the highest density of bacteria are found, in part because it just, you know, it happens to be my favorite organ system, and in part because you all probably just had dinner, and so it's a particularly good time to be thinking about what's actually going on down there while you're digesting. So, there are bacteria in the gut. You all are aware of that. What bacteria are in the gut? Does the inside of your gut just look like a little microcosm of all the bacteria on the planet? Are there particular bacteria that particularly like to live in your gut that are more highly enriched there than there are anywhere else? Well, from the way I asked that question, you could probably guess that the answer is the second thing, that if you compare all the bacteria on the planet, their abundance and their diversity, to the bacteria that come out of your back end every day, those are different subsets. In particular, the bacteria that are found in the gut are relatively low diversity compared to the overall bacterial tree. So what we're looking at here are two more trees where I've gotten rid of the archaea and the eukaryotes because we're not going to pay attention to them anymore. And here we're just looking at the bacteria. And this shows a slightly more elaborate version of the bacterial branch I showed you on the previous slide, where here the size of these branches reflects the number of different species that have been identified that falls into each of these groups. So for example, the firmicutes up here are a pretty abundant phylum of bacteria where you have lots and lots of different species that fall into that branch. Question?
Can you put the microphone more towards your face?
Oh, I'm sorry.
Not a problem.
Okay. Okay, so this is all of the bacteria, and these have been assembled by, again, taking random samples from all over the world, lots of different kinds of environments. And then this is a similar plot showing the bacteria that are found in the gut. So most of them, as reflected by these big objects, are a class called Bacteroidetes and a class called firmicutes. And there's a few other things, for example, proteobacteria, gamma proteobacteria over here. This is E. coli and all of E. coli's friends. We like it because it's very easy to culture, but it's actually a fairly small minority of the bacteria in your gut. Most of them are these other two classes. Now, you can do the same experiment. Instead of just taking the bacteria and measuring the ribosomal RNA sequences from the gut, you can do this, of course, for any part of the body. And one of the things that's been going on in Dave Relman's lab here at Stanford, as well as a lot of other labs around the world, has been just to do surveys, to do a census, if you will, of who lives where and how many of everything is where. And this is a summary slide from one of David's reviews, just showing the approximate numbers and then the phylum distributions of bacteria from different parts of the human body. So here's the gut. This over here represents the Bacteroidetes, and this over here represents the firmicutes. You see it's mostly those with a little smattering of some other things. But if you were to culture bacteria from the skin, you'd see something very different. Most of those are actinobacteria. You do have some of the firmicutes here, but then also more proteobacteria, relatively few of the Bacteroidetes. If you go in the mouth, you see a different population. If you go in the esophagus or the stomach, so higher up in the gastrointestinal tract, you'll see different populations. And in the vagina in females, there's also a very rich microbial environment there that, again, is completely distinct from the skin or from any other part of the body. So how many different bacteria are there? That's really not known. If you were to ask a microbiologist 50 years ago, they would say, oh, there's a few dozen. And the reason they would say that is because if you take a sample from any one of these sites and then you try to grow bacterial colonies out of these samples in the laboratory, it's only a very small minority of bacteria that can be cultured in that way. Most of the bacteria that are being tracked now using this ribosomal RNA sequencing technique are bacteria that we've never been able to grow, we've never been able to isolate. We may be able to see them in the microscope as individual cells, but we can't make a whole colony of them, and we can't really analyze their metabolism or their behavior in sort of a classical bacteriological way. The sequencing technology has been getting better and better, and Mike Snyder mentioned a little bit of this last week. The same kinds of high-throughput sequencing that have been applied to human genome sequencing also can be applied much more easily to sequencing of bacterial genomes and bacterial RNA. So as these technologies are getting better and better, the estimate for the number of different bacterial species that are in there keeps going up and up. So as of about 10 years ago, there were only about 400 species in there. As of this year, there's probably about 30,000 species in there. I don't know how much bigger that number is going to keep going. But although there's lots of different species, that doesn't mean they're all equally represented. So there are a few where there's a lot of them, and then there's a few where there may only be, one or two. Some of them are permanent residents in your body, and some of them are transients that will come through or will colonize you briefly and then will go away. And we're just beginning to understand the dynamics of those populations as they come and go. It really is like an ecosystem. And so you can think of it as being like a forest or a kelp forest under the water if you're a scuba diver. There are things that are there all the time and things that come and go and things that are present in different numbers. And we're just beginning to get familiar with what all those things are. So thinking about the intestinal ecosystem, I'd like to briefly mention the work of one of my junior colleagues in the Department of Microbiology and Immunology, Justin Sonnenberg. Justin's very interested in how the bacteria that are living in the intestinal ecosystem interact with the human cells. And in particular, if you think about the bacteria that live in your gut, they really have a pretty good deal. It's nice and warm in there all the time. It's nice and moist. And 2 or 3 times a day, every day, you deliver a great bolus of food right to where they're sitting. So you take good care of the bacteria in the gut, but you can choose to feed them different things. And as you might expect, if there's some bacteria in your gut that are extremely good at digesting plant matter, but you're somebody who never eats vegetables, you only ever eat meat, those bacteria are going to be very unhappy. Now conversely, if there's some bacteria that are extremely good at degrading protein, but you only eat meat, those bacteria are going to be happy. So depending on who you are and what you eat, you're going to have different populations in your bacteria that are responding to the signals that you give them in terms of what you feed them. Now, besides that selection that's operating on all those bacteria in your gut, there's also going to be interactions between different bacteria in the gut, both microbes of the same species that may cooperate with one another, and microbes of different species that may either cooperate or compete with one another. And at the same time, different individual people are going to be making different kinds of signaling molecules, different kinds of immunological reactions that will interact with the bacteria differently. And one other particular component of the intestinal ecosystem, which I think doesn't get enough appreciation, is the mucus. Now, we, sometimes out in the world we think of mucus as being kind of disgusting, but actually mucus is one of the most miraculous substances produced by your body. You produce it all over the place in your lungs and in your intestine, as well as in your nose when you have a cold. And it's actually a critical component of the functioning of all of these organs. So in the intestine, the secretion of mucus, which is long-chain polysaccharides that are very hygroscopic, that keeps everything nice and slick and moving along. And in fact, people who have deficiencies in producing mucus have very severe difficulties with their gastrointestinal function because they lack that normal lubrication. Another good function of the mucus is to keep the bacteria, particularly in the lower intestine where they're very numerous, to keep them off the surfaces of the host cells so that they can do their work in the lumen of the gut but not actually have direct contact with the cells. So it's a little bit of a protective function as well. And finally, this mucus that you make is made up of, I said, polysaccharides, that is long chains of sugar. And there are some bacteria in the gut that are specifically good at digesting those sugars. So you're feeding the bacteria in your gut, not just by the food that actually goes in your mouth, but also by the sugars that you're secreting from your intestinal cells. And this is all presumably done in a way where everything is reasonably balanced and deliberate in order to maintain health over the many years, many decades of a human life. Now, in Justin's group, they take two different approaches to trying to understand what's going on in the intestinal ecosystem. One is to embrace the complexity and try to look at all of the different organisms that are in there. And the other is to try to simplify the system and see what's the simplest way we can set up in the laboratory to study this kind of thing by putting in defined microbial communities. So I'll just mention a couple of examples of different ways to think about that. So starting with the thing about embracing the complexity. So let's actually now look at some bacteria in the gut. This is an electron micrograph of a part of the small intestine where the donor, in this case a mouse, gave its intestine, which was then filleted open, and then most of the contents were scraped off. And after most of the contents were scraped off, what you can see left behind here, these little fingers that are sticking up, these are what are called intestinal villi. And these are projections of the epithelium in the intestine, that is, of the layer of cells in the intestine that actually do the work of absorbing the food. So there's one cell, there's one cell, there's one cell. They're all organized together in these beautiful little structures. Now this stuff over here that's lining up here, this is the mucus. And most of the mucus has been scraped off so you can see the villi, but under normal circumstances, the whole thing is covered with this. And if you look at the mucus very closely, you can see these little rod-shaped bodies in there, and those are the bacteria that are associated with the mucus. Now we have here a couple more views. This again is a close-up view where you can see the bacteria a little bit better. And this is scanning electron micrographs. You can also look at this in a section of an electron micrograph. And this is a little bit more accurate because here the gut contents have not been removed. So over here on the right half, these are human cells. These are the little villi. And you can probably see these circles of little nuclei all in a row, which are actually the outside of the villi with the intestinal epithelial cells all lined up to do the nutrient absorption. And then this stuff over here, this is the luminal contents of the gut. So this is the food that's being in the process of being digested. And we're down here now in the lower part of the gut. So this is a lot of probably mostly plant matter because this is a mouse. Again, more plant matter that's filled with bacteria that's digesting it. And these two things here are gas bubbles. So they're incipient farts. So the way to think about what's going on in here is you have plant polysaccharides that the mouse is eating. They come down in there and then they're degraded and fermented by the bacteria. And the bacteria release metabolic byproducts, including short-chain fatty acids and acetate and butyrate and a lot of other good things that then can actually be absorbed by the human and then used for food. So there's a lot of plant material that you eat that you have no enzymes in your genome that can digest them. But the cells, the bacteria that live in your gut, particularly the Bacteroidetes, these guys specialize in breaking down very exotic kinds of plant sugars. and then they'll give them to you. And then in the slide that I got from Justin, he also likes to point out that in these micrographs, of course, everything is dead and fixed, but in reality, all of this stuff is very dynamic, like a kelp forest. Everything is moving around, and there's a constant current or flow of material past all these things. So for the bacteria that live in there, they have to be able to maintain themselves there, replicate, and also hold on so that they don't get washed out along with the progression of food through the gut. So again, thinking about the food digestion, you bring in the carbohydrates. Those go through primary fermenters in the gut, which are bacteria that then generate a bunch of small molecules that can be used by other cells. There's also some other bacteria specifically that produce lactic acid, some other bacteria specifically that produce butyrate. Butyrate is something actually that can be used directly by the human host cells over here. It can be taken up and used as a nutrient source. There's things that produce acetate. Then you can take some of these other small molecules and feed them to the acetogenes that will produce more acetate, and that can then go and feed the epithelial cells. But then there's whole other layers here. So after these primary fermenters have started breaking down the sugar, they can feed some of the components to other things, methanogens. These are largely actually archaea that make methane, which smells bad. So you know how some people's farts are a little bit smellier than others? I mean, it's an observation, it's not a judgment. That's because some people have higher populations of methanogens in their guts than others, and it's just one of those natural sources of human variation. There's also sulfate reducers, and actually whereas all these other things that are on this slide are things that are good for the human cells, like butyrate and acetate that you can use as a food source, hydrogen sulfide is actually damaging to human cells. So there's got to be some sort of balance or some sort of give and take to make sure that everything stays intact. So one of the reasons that Justin and other people in this field are studying this is just to try to understand enough about the interaction between the metabolism of the bacteria and the metabolism that we're trying to do so that we can ideally manage the community and manipulate it to maximize our health. Now, another way to think about the diversity is to think about the question of, so the metabolic slide that I just showed you sort of suggested, in your gut, you meaning any of the 200 people in this room, in your gut, this is what's going on. But really, if you were to take you personally there, compared to the person sitting next to you, you were looking in your gut, there may be slightly different things going on depending on what you're eating and all of those other sorts of variations. So Dave was interested in this and started off just by looking at different kinds of animals to see, if you take a human or you take a mouse or you take a rat or you take a rabbit, how similar are the bacteria in the guts of these different mammals. And this shows this down here. These are 4 different animals. This is human, mouse, cow, and pig. And this is the overall bacterial tree that I've shown you a couple of times before to get just a slightly different representation. And down here, any place where you see a different color, that's where you see a particular bacterial species that's found only in one of these animals and not in the others. So what I hope you can appreciate is that the overall pattern is pretty similar between the sort of consensus pattern here and each of these individual animals. But the details of the pattern are different. That is, as you get out into the sort of branches or twigs of the tree, that's where you start seeing a lot of variation. He followed this up then with what I think is a really spectacular experiment of just taking 3 different human beings, perfectly average normal human beings, human volunteers, and then enumerating the species that were found in their feces. And here, again, you can see the overall shape of the trees is very similar, but even just among 3 different humans, you see a lot of colors here that shows that there's bacterial diversity as you go from one person to another.
What about within individuals?
Within an individual over time, yeah, that's a terrific question. So the place where that's been studied the best is when you have an individual who is sick. And there, it's very clear if you have a bacterial infection in the gut, there's massive changes to take place in the whole population. It can take a very long time until the population gets back to normal. Now, you'd also want to know, just as you're walking around like from day to day and week to week, how much variation there is. And that has not been done yet very carefully in adults, but it has been done in babies. And I'll show you the babies on the next slide. Yep.
Question about some foods like yogurt advertise that they have live cultures in them.
Yeah.
Comment on how that would affect the situation in the guts.
Yeah, so the question was for foods that have live bacterial cultures in them, and yogurt is one of the most obvious examples. How does that actually affect the microbiota population in the gut? So that, again, is someplace where I would say the really rigorous scientific work hasn't yet been done. What is certainly clear is that when you take antibiotics, you get massive loss of populations of bacteria in the gut. And then there is, at least reasonably good evidence that eating yogurt sort of helps repopulate that. What's not at all clear is that the bacteria that are in the yogurt actually end up staying in your gut for any length of time. But there is apparent clinical benefit, at least for some people, of eating the yogurt. So what it might be is that although the yogurt that you eat, the bacteria there are transients, they don't stay around, they may still sort of change this dynamic food web of all this metabolism in a way that then encourages the regrowth of the normal flora. But that's extremely controversial. I mean, that's the sort of thing where, you know, there's obviously companies who are interested in things like selling probiotics that have a very specific desire in having very specific scientific results come out. But I haven't personally yet seen anything published, you know, from basic science research that would really convince me that we understand what's going on. Was there another question? Okay. Okay, well, let's talk about the babies. So this is a terrific experiment. And what inspired this was, you know, this kind of work saying, okay, all these different adults, you know, they have different microbiota in their guts. So how did that happen? When we're born, we're sterile. And then we acquire bacteria on all of our different surfaces over time. So how do we acquire them? And how long does it take before you set up a stable population? And when do people become different? Do they start off different and then get more similar, or start off similar and then get more different? Question?
You're sterile, I mean, in the baby gut, there is no bacteria?
No bacteria in the baby gut. Question? That was the same question. Yeah, when we're born, we're sterile. Isn't that true? Yeah? Okay. I had to ask the real doctor, but yeah. But not for long. So as soon as you come out, you know, you start breathing stuff starts falling on your skin. As soon as you eat something, you got bacteria coming in there. So you get colonized. So this was a study, no, seriously, that's the technical term. So this was a study done by Dave Relman together with another of my colleagues, Pat Brown, where their graduate student, Channa Palmer, she went around Beckman Center, which is just across the street here, and looked around for women who were pregnant and asked those women if they would be interested in participating in this study. This is a research building filled with graduate students and postdocs, and of course all of them said, yes, I'd be interested in participating in your study. And what they did was, for all of these women who were about to have babies, they gave them a very, very large number of plastic conical tubes, and they asked them for the first year of their baby's life to keep a little sample of poop. For the first couple of weeks, to keep a little sample of poop every day. For the first couple of months, to keep a sample of poop, you know, a few times a week. And then, you know, keep doing this for the first year of the baby's life, so you get just a whole record of everything that came out the baby's back end. Now, at the same time, because these are families, they also wanted to know what's going on in the mother, because it's an obvious question is, you know, whether the baby, when it gets its bacteria, just gets, you know, what the mother gave it. Then they also want samples from the father, and they also want samples from the siblings. So actually, you know, one of my postdoctoral fellows was one of the participants in the study, and boy, their freezer was full of full of little tubes of poop. But you know, these, as I said, they're graduate students and postdocs. They were very happy to participate. So what Channa Palmer did then was go around and collect all of these samples, and you know, and then go enumerate what all the bacteria were that were in there. And there's a couple of different ways of looking at this data. This over here just shows the total numbers of bacteria that are in the baby guts as time goes on. So here, for example, this is right after day 1, and then this day, or sorry, 2, 5, 10, 20, 50, 200. So time is going faster as we go along here. So you start off sterile, that is, you can't detect any bacteria. Then over the first few days or few weeks, the number of bacteria comes up to what seems to be a steady state level. But one thing that's very striking in the babies is every so often they just crash. So you get a sample out where there's basically no bacteria in there, at least no bacteria you can count. And that happened, one of these children, I think it was this one, actually was sick and was given antibiotics. And so the crash came right after the antibiotics, so that makes sense. But a bunch of these other babies, they would crash for no apparent reason, and then the bacteria would bounce right back. So it's not all clear what's going on there. And then it starts to look even more interesting when you actually look specifically at which bacterial species are present. So in these graphs, the identity of the colors is not important, but the point is that each of these colors shows the relative representation of a particular group of bacteria, sort of particular phylum of bacteria. And what you see is they all are just all over the map. You know, things go up and down. No two babies look alike. They're colonized in what the author said were a seemingly chaotic way, where stuff comes and goes, you don't know why. Interestingly enough, by the time you get to the end, by the time you get to 1 year, then all the babies start looking more or less the same, and all the babies start looking like adults. There was no correspondence between what colonized the babies and what was in any of their other family members. But each one of them sort of went through this whole drama of like interacting with the world and getting stuff and rejecting stuff and trying again. And then it all settled down at the end of a year. Yep?
Is it the same for breastfed
I don't recall. I think they did have some of each in the sample, but I don't remember if they had enough that they could draw any firm conclusions about it. But I believe the majority of these babies were breastfed. Yep.
How do you explain these babies that got high counts at birth, like 5, 10?
Yeah, so that's not actually birth, that's 1 day after birth. And 1 day is a reasonably long time. But I don't know if there's anything special about those particular babies. At least it wasn't described in the paper. But that's just sort of part of the, the overall chaotic pattern. So you could say sort of the general rule is you start off low and then go high. But yeah, you're absolutely right. There are few. So that was high on 1 day and low on the second day. Who knows what happened there. Also remember, these are parents who are collecting the samples from the babies, so they're not necessarily done under perfectly reproducible conditions. So that might contribute, but I don't know. Yep.
How does the bacteria get into the gut? Can it survive the HCl in the stomach?
Yeah, so the question was how the bacteria get all the way down to the gut if they have to go through the acid in the stomach. So a lot of bacteria are very acid resistant, and some bacteria even secrete things like, secrete specific enzymes that will overcome the acid. And even if you're not an acid-resistant bacterium, the thing about bacteria, sort of their strategy in life is to say, okay, we'll just make lots of ourselves, and if 1 out of 10 to the 5th survive, that's good enough. So it seems like a lot of that is probably just, occasional good luck of the occasional ones who make it through the acid.
It's very low in newborns.
Oh, and the pediatrician, Dr. Pizzo, just told me that the acidity in the stomach is very low in newborns. So that probably helps things go a little faster.
So one way to repopulate the bacteria when there's a crash, or say an adult, would be literally to
Constant supply.
Oh, so if an adult takes an antacid and then tries to repopulate, would that help?
feces.
Yeah, no, that's actually.
Yeah, we'll talk about that in a minute. That actually is a very good way to repopulate.
Yeah.
Sorry?
We can talk about it later.
Yeah, okay, well, we'll talk about that later, but hold that thought about eating feces. That certainly is about to become relevant. Okay, yep? What is the typical lifespan of a bacterium? So that is a question that I think cannot actually be answered because bacteria replicate by binary fission for the most part. So if you have one bacterium, it divides in two, you don't have that same bacterium anymore, but does that mean it's alive or dead? Now, some of them are going to die, but they're going to die because of circumstances or because of competition. So the average replication rate of a bacterium can be anywhere from 8 minutes in the laboratory to in the gut, the average is probably on the order of about 3 hours. But then there's some bacteria, there's actually this fantastic paper that came out last year where there's this place in Antarctica that's called Blood Falls, where you see this bright red water pouring out through a crack in the ice. And it turns out what happened there was when all the ice was shifting around, something like 3 million years ago, it trapped a pocket of seawater. There's some bacteria in there, and they've just been in there ever since. And they've been evolving as a community where they trade off their nutrients with one another. There's no new stuff coming in, but they just keep going. And they're red because there's an iron that plays an important part in the oxidation of that. And so these guys, they actually calculated how frequently they divide. They divide once a year. But then, how long do the individuals live before they die is going to depend on the circumstance. So it's highly variable. Yep.
Would there be any association with the crashes between a baby being fed with mother's milk as against the bottled milk or some other type?
So the question was if there were environmental causes of the crashes, for example, a change in food source like from breast milk to bottled milk. That was not described in this paper. I don't know how carefully they documented exactly what the babies were eating. So I'm not sure that that information was available from this particular study.
It says little similarity except for twins. What were the similarities in twins?
Oh, yeah. So I wanted to point that out here. Number 13 and number 14 were identical twins. So if you look at the overall patterns here, sort of what comes and goes when, and you can also do this quantitatively if you want, these two babies are more similar to each other than they are to anybody else. Now, in part, that might be because they're genetically identical, and there could be some component of the host genome that influences the way these things go. Of course, most of the time, they're lying in the same crib. So it'd be really interesting to have fraternal twins to compare to the identical twins. But they just got lucky, that they got identical twins in the sample. But that's the sort of thing you'd want a bigger study to know for sure. Yeah?
When babies crash, do some of them not rebuild their system? And does that contribute to something like sudden infant death syndrome?
Who? So in this particular sample, every one of the babies that crashed rebuilt before the next sample came around. So my guess is that kind of thing is happening all the time. We just don't notice it. I don't know if the cause of sudden infant death syndrome is, yeah. Okay, so the authority says no relationship. Yep.
Are the microbes evolving in the gut, or are they getting selected from the environment?
They are, well, okay, so let's do a little calculation. So bacteria in the gut are going to divide about once every couple of hours. If you actually count then how many generations that's been, and then you say, well, how many human generations that would be? If you do this calculation, you say human generation is 20 years, bacterial generation is 2 hours. That's equivalent in the gut for the bacteria to have evolved as far as humans have evolved in 6 million years. Do you know what happened to us 6 million years ago? We shared a common ancestor with chimpanzees. So there's a lot of evolution, there's a lot of time, there's a lot of mutation, and also, as we'll see when we finally get to the part about sex, there's a lot of swapping of DNA that goes on in there. So there's constant repopulation from the food, and then there's lots and lots of stuff going on inside. Yep.
The popular concept of detox, what effect does that have on gut bacteria, if any?
I'm not sure I'm familiar with exactly what's meant by detox.
Purges.
Purges get rid of bacteria.
It drops you by logarithmic. So
Yeah.
if you
Why
look-
would you do that? Sorry.
Colonoscopy.
Oh, okay, okay. So if you get a colonoscopy, then you do it, I guess, because you want things to be clean, and it's hard to see if it's full of stuff.
But it is both transient and it's not absolute. So if you actually monitor bacterial cultures, as I did once in people who are taking antibiotics, you just reduce it down to a certain threshold. They'll eventually grow. There's so many of them,
Yeah.
obviously.
Yeah, but it has been shown. So there have been some studies of this kind done deliberately where human volunteers would take Cipro or would take some antibiotic, and then the microbiota would be tracked. And there's changes that last for months. You know, it takes a long time for everything to really get back to normal. Is there
That's
another
where it
question?
affects the
Yep.
air roots in the animal.
Just in general, the bacteria are sitting there digesting food that we can't. What good does that do us?
Oh, they feed it to us. They give us acetate, they give us butyrate, they give us their metabolic byproducts we take up and use.
So we eat their poop.
We eat their poop. Yes, that's exactly right. Happy to clear that up. All right. So there's all these microbiota in there. They get established. What effect does this have then on the behavior of the human? Now, as we mentioned, one thing is they actually provide us with some nutrients. But it's also conceivable that because you have all this stuff going on in there, you have all these bacteria that are being recognized by the immune system, they could have more profound effects. So one of the approaches that people have taken to study the overall effects of gut microbiota on the development of the gut, and in principle you could do this also with other tissues, was pioneered by Jeff Gordon at Washington University in St. Louis, which is where Justin did his postdoctoral work. And the approach is to make mice that are germ-free. So as I mentioned, when we're born as babies, we're sterile. We start off being sterile. And the same is true of mice. So if you take a mouse that's pregnant and you clean off the surface and you put it into an isolation chamber, and then you give the mouse a cesarean section, take the little pups, and you're very, very careful to keep them sterile, you can actually establish a colony of sterile germ-free mice. And these guys have to live in a bubble the whole time. There's sterilizing filters to stop anything from coming out from the inside. You have to autoclave all of their food and water to get rid of all the bacteria, and you have to be very careful. But if you do this properly, you can raise whole colonies of mice where there's no bacteria on them anywhere. And that's illustrated over here. This is a plot showing the position in the gastrointestinal tract from the ileum, right when you come out of the stomach, down through the cecum, the parts of the colon, down to the rectum and the feces, showing the population of different bacteria in different locations there for a normal mouse. And then this is the same plot for one of these germ-free mice. There's just nothing in there at all. But once you have a completely germ-free mouse, then you can first off look and see, well, what happens to the germ-free mouse? Is it a problem for it not to have any bacteria in the gut? And then the second kind of experiment you can do is you can deliver only specific bacteria to that germ-free mouse. So instead of giving it the whole 30,000 species or whatever that it normally has, you can give it one bacterial species, or you can give it two bacterial species. This is called a notobiotic experiment, where notobiotic means known life. That is, you know everything that's in there. There's the mouse, and then there's only what you gave it. So there's obviously a lot of very interesting kinds of experiments you can do with this. So first off, I just want to tell you a little bit about what goes on with the germ-free mice in terms of their digestion. One thing is they have a real big problem digesting plant matter. And mice are vegetarians, so the mouse chow that you give them is plant matter. And normally, all of those polysaccharides are broken down to make these smaller short-chain fatty acids and acetate and butyrate and things like that, the bacterial poop, which then goes to feed the mouse. And this is a part of the small intestine called the cecum. If you take a normal mouse, that's what the cecum looks like. And if you take a germ-free mouse, that's what the cecum looks like, because it's totally clogged up with undigested polysaccharides. Now, obviously, there are ethical reasons why this experiment cannot be performed in humans, but it seems very likely that the same thing would happen to us, that if we couldn't have the bacteria helping us digest that stuff, everything would get clogged up. Question?
It's actually a comment. In transplant setting, we are becoming very good about doing gut decontamination before a transplant because you want to avoid some problems. And there is an issue, especially if you have graft-versus-host disease, that you in the gut eventually afterwards, and you also lose the villi, that the lack of diversity of bacteria cause a numerous amount of issues. So we are involved in a study that we do freeze a lot of poop too. But we are pretty much trying to establish that the lack of diversity in a human gut, it's very detrimental for our health. And we are trying to come up with good ways of recontamination of the gut that will be effective and therapeutic to avoid even further issues.
That's very interesting. I'm just going to repeat the comment for those of you who couldn't hear it. This student here is involved in gut transplantation, and normally if you're going to do a transplant into a recipient person, you want to decontaminate the gut, clean all the stuff off of it.
Just a correction. I do bone marrow transplantation.
Oh, bone marrow transplantation.
Yeah. So, but we need a clean gut as a way to
prevent
some stuff. So we do have those same issues. So the transplant is not intestinal. Yeah.
The transplant is not, okay. But still you need to decontaminate the gut in the context of this. And the observation in these human patients is that if the gut is very clean, then that actually causes big problems in terms of digestion, including loss of the villi, loss of the normal structures. So they're trying now to figure out how to recontaminate the gut in a responsible way to stop those problems from happening. So that's very interesting. Yep.
What about just saving some of your own and then reintroducing it?
Yeah, so in principle you can certainly do that, is save some of your own poop and then yeah, and that's done. Yeah, that's done not that uncommonly. Yep.
Is Crohn's disease, is that due to lack of something in the gut?
Crohn's disease. Yes.
It's an inflammatory bowel disease.
Okay, so the actual doctors are telling me that it's an inflammatory bowel disease, so caused by an autoimmune reaction. Okay, so it is. So I think that in Crohn's disease patients, the gut microbiota is different, but that is, I believe, a consequence of the inflammation rather than a cause of the disease. Yeah. Yep.
How long would the notobiotic mice live compared to normally grown-up?
Yeah, so the question is, how long do the notobiotic animals live? Do they have a shortened lifespan or extended lifespan? I don't know the answer to that. It's comparable, but it may be a little bit shorter. Yep.
Simplistic question. So here you're saying that all the bacteria, they can break down the food and that's what we consume. So that's a good solution for obesity or weight reduction problems?
Yeah, so let me hold off on that question because I'm going to show you data about microbiota and obesity in just a minute. Okay? Right, okay, anyway, so here we are. So having a sterile gut is not good, as was told to us in the context of transplant patients, and then also this has been shown in many different ways in the notobiotic animals. So not only do they pile up the plant matter because they can't digest it, but then actually the intestinal cells of the host, of the mouse, they're different, and they're different in detrimental ways. So for example, in order to absorb the nutrients from your food, you have the intestinal epithelial cells that actually transport the molecules, and they deliver them into the bloodstream. So there have to be blood vessels that grow up into these villi, into these little fingers, to receive the nutrients. And these are sections through villi from a germ-free mouse, and then from a normal mouse, and then from a germ-free mouse that's been given just a specific subset of the normal bacteria. What I hope you can see here is the blood vessels that are labeled in green in the germ-free mouse are very sparse compared to this nice elaboration of branched blood vessels that you see in a mouse that either has a full complement of the normal microbiota or in one that's been reconstituted with just choice microbiota. So you need to have the bacteria in there to encourage the blood vessel growth within the villi. Another thing that's been demonstrated is that in the germ-free mouse, there's a change in gene expression, specifically of proteins that are involved in food metabolism in those intestinal epithelial cells. So the way that you get sugar in from the gut is through specific transporters. One of them here is called sGLUT1 that transport glucose and some other sugars. And in the absence of any bacteria in there, the expression of this transporter is greatly reduced. So you can't take up even the sugars that you could digest yourself. You can't take them up as efficiently if you don't have any bacteria in there. The exact mechanisms by which the bacteria signal to the gut to change their gene expression patterns and to change the angiogenesis, that's still under investigation. But the fact that they do have these effects, I believe, is well established. And this is just to remind me to mention that people are also doing this kind of thing in fish and also in chickens. It's actually extremely easy to make a gnotobiotic chicken because they come in eggs. So all you have to do is scrub off the surface of the egg, put it in a germ-free environment, and the chicken stays germ-free. And at least among fish, chickens, and mice, these same sorts of interactions all seem to be conserved. Okay, so another sort of interesting effect on the host has to do with the immune system and has to do with the host metabolism. And this gets back to this question about obesity. So if you look at, again, conventionally raised mice versus germ-free mice, and you compare here, these are the epididymal fat pads, so this is equivalent to the sort of jelly roll on the mice. These are the germ-free, and these are either conventionally raised or these are ones where things have been added back. So these germ-free mice eat more, but they weigh less than normal mice. Now, this doesn't mean they're more healthy. It actually means they have difficulty absorbing their food. Now, you might ask what connection this has or may have to human obesity. So, of course, it's a very obvious experiment to look at changes in microbiota for either humans that are obese or lean or for mice that genetically are driven to be obese or lean. Now, these are not germ-free, but this is either normal mice or mice that have a genetic deficiency that causes them to be obese, or for the heterozygotes. And here we're looking just at the two major populations, the firmicutes and the Bacteroidetes. You see this is the normal ratio between the two, but in the obese mice, you have a lot more firmicutes than you should. Now, interestingly, if you look at humans, the overall ratio between the firmicutes and the Bacteroidetes is not the same. But here on this side, this is lean humans, and here on this side, this is obese humans. And you see there is, again, a very similar change in terms of the ratio of these two things. Now, one thing that's really, really interesting in this particular study that was published in Nature in 2006 is that in this case, the human volunteers went on a diet and lost a substantial amount of weight over a fairly brief period of time, over about a year. And actually, over time, as these people spent more and more weeks on this low-calorie diet to lose weight, you see the ratio became closer and closer to normal, closer and closer to the ratio of the lean people. Now, what of this is cause and what is effect is still extremely controversial. To get a cause and effect in humans, as you're aware, is extremely difficult. But in the gnotobiotic system, you can do a very clever experiment to see whether the difference between leanness and obesity is causing the change in the microbiota or is a consequence of the change in the microbiota. So here's the experiment. There are these mice that are genetically altered to be obese. And this shows one of these obese mice next to one of its normal litter mates. So they're very big. They're 2 or 3 times the weight of their normal litter mates. And we talked before briefly about eating poop. That can never be revisited too frequently. Mice actually eat each other's poop and their own poop just as part of their normal habits. This is very common among rodents. If any of you have ever had a pet rabbit or a pet rat or a pet guinea pig, you'll see them all the time, you know, sort of munching on the raisins. It's actually part of how they keep their digestive health is to eat feces. So, so this experiment is done where you take these germ-free mice that are raised in the gnotobiotic facility and you feed them the feces of other mice, which they're very happy to eat because they like it. And you can feed them either feces that come from a wild-type normal mouse that's out in the world and has normal microbiota, or from one of these obese mice that is genetically altered. And what is found is shown here, which is really quite astonishing. Remember, the germ-free mice have problems absorbing their food. So if you feed them poop from other wild-type mice, they'll put on weight because they'll get a microbiota that'll be able to absorb food. They put on about 20 to 25% of their body fat after they've been colonized with the normal mouse's poop. But if you take the poop from an obese mouse, you do exactly the same experiment, they put on almost twice as much body fat. So there is something about the identity of this microbiota that is able to determine the outcome as far as the metabolism of the recipient. Yep.
What about the lifespan of the fat mouse?
What about the lifespan of the fat mouse? So in general, fat mice don't live as long as lean mice, but in this particular experiment of the transplant, I'm not sure that it was carried out to look at the lifespan. At least that wasn't reported in the first paper.
What genes were altered in the obese mice?
So the obese mouse has a mutation in a gene called leptin, which is a signaling molecule that comes from the fat cells from the adipocytes and then communicates to the rest of the body when the mouse is full. And it has effects both on how much the mouse eats and also on the mouse's activity. So a mouse that has this mutation never feels full. It eats all the time, and it doesn't get very much exercise.
Does it affect its immune system?
Does it affect its immune system? I don't know. It might, but I don't know.
The opposite is true. Malnourishment affects your immune system.
Okay, so I'm told that malnourishment affects your immune system, which makes sense. Certainly you'll be more susceptible to infection if you're malnourished. But whether obesity per se or leptin deficiency affects the immune system, I don't know. Was there another question? Yep.
So when you feed it the waste from the other mice and the plants, how do you make sure it is germ-free? Because these mice are supposed to be
After you feed them the poop, they're not germ-free anymore. It's just they're gnotobiotic in the sense that you know what you gave them. So you have to keep these mice, you keep in a bubble that's separate from these mice and that's separate from all your other mice. So you give each of those little colonies, you give only a certain kind of poop. Yep.
Did the mice on the right reestablish a normal genome, or a normal bacterial colony, like the ones on the left, after a while, after they stopped eating the poo of the?
How long does this effect persist? I don't know how long the effect persists. Yeah, I mean, you could see either way, that because it's a sort of self-perpetuating state, it could be something that you get into that state and you can't get out of it, or it could be that over time there'll be a gradual renormalization. Now, I would bet, well, I don't know. I don't know which way I would bet. I could see it going either way. It wasn't reported in the first study. Yep.
So I'm still confused about the effect here, because supposedly the fatness of the fat mouse is caused by the crazy leptin.
That's right.
Whereas the fatness of the normal mouse is caused by the same bacterial profile as in the leptin mouse.
That's right. So yeah, so this is why this is such an interesting result, because what it suggests is, we know the reason for the obesity of the obese mouse. It's because of this leptin mutation. But the obesity, the metabolic changes associated with obesity, then are some way altering its microbiota, where then the microbiota get into some state where they then can carry that information to another mouse without carrying the mutation, right? So there's feedback in both directions, from the host to the microbiota and from the microbiota to the host. And that's what's demonstrated by this experiment. The mechanism, as I said, how those bacteria talk to the host and cause it to become obese, mechanism is not yet known. Yeah?
So the mouse that became fatter, did they try feeding that species of lean mice?
Oh, to see if that would fix it. Again, not in this original paper, but that's the experiment I would do next, and I bet they're in the process of doing that. Yep.
So if you put in order of increasing weight, the different mice involved here, how would it look?
Oh yeah, the genetically obese mice are fatter than these guys. Right. It's yeah, and this I believe they get back to pretty much a normal weight. So the germ-free mice, are lowest. The normal mice and the reconstituted mice are moderate. Then the ones reconstituted with the microbiota from the obese mouse, and then the obese mouse is the biggest. Okay. All right. Yeah, so this is in mice. Somebody actually asked about Crohn's disease, and for other kinds of bowel diseases as well, including inflammatory bowel disease, there are observations that there are changes in the microbiota that are associated with this. Here, this just shows, again, one of these plots of the different populations in different parts of the gastrointestinal tract. And then for a mouse that's been given a form of inflammatory bowel disease, you can see everything is different really pretty much all the way down. So there again, it's very hard to distinguish between cause and effect in those things, but it could partly be that the persistence in the altered microbiota is part of the reason these chronic diseases are so hard to treat. So, okay, so that is the first third of my talk. You guys are great, actually. It's been really fun. So Yeah, so I think I'll just keep going and we'll just stop when we stop. And please keep asking questions because it's really enjoyable for me, and I hope for all of you too. So, so the next two parts are going to be about how disease-causing microorganisms differ from the benign or helpful organisms that normally inhabit us. And then the third part was going to be about human cells interact with the helpful and harmful microorganisms. So let's just plow on ahead. So we've been talking so far tonight about the benign organisms that live all over us and in us all of the time, and how they help digest our food and do all these other good things. But there's lots of them, and we're most of the time healthy, walking around with all of these passengers. But it is the case that there are some microorganisms that cause disease very specifically, even in healthy adults. And for example, I said there's, in your gut, there's on the order of 10 to the 11th bacteria per gram of gut contents. But if you take a glass of water that has 100 organisms, so 10 to the 2 as opposed to 10 to the 11th, 100 organisms of Shigella flexneri, and you drink that glass of water, you will have dysentery, you will have bloody diarrhea. So there's something about those very small numbers of very bad bacteria that are able to somehow do something profoundly different from what all of the benign inhabitants that we normally have can do. So that sort of raises a question, an interesting question, and one where a lot of the field of bacterial pathogenesis has been focused for the last couple of decades about what's different between Shigella and its relative E. coli that lives in the gut normally and happily. So I'd like to start this part just by reminding you that there are lots of different kinds of microorganisms that cause diseases. For bacterial diseases, there are things that come in epidemics or plagues, like cholera and bubonic plague, but we can also have viral plagues like AIDS or like influenza. There are diseases both on the bacterial sides and the viral sides that have been with us for a very long time, like tuberculosis or leprosy or polio. There are some that have been pretty much wiped out on both sides, largely by the advent of vaccines. So scarlet fever used to be a very common cause of death among children and is now pretty much gone because we have a very effective vaccine. Similarly, smallpox has been completely eliminated in the wild on this planet by the use of a vaccine. Now, you can get bacterial diseases different ways. For example, some you get from animal bites, Lyme disease and rabies, or bacterial and viral diseases from animal bites. Some you get sexually transmitted diseases. So syphilis, gonorrhea, and chlamydia are all bacterial sexually transmitted diseases, but herpes and hepatitis are viral sexually transmitted diseases. There's some diseases, again, on both sides that you hear about all the time, but you will never get. That includes flesh-eating bacteria and Ebola. I promise you won't get it. On the other hand, there's bacterial and viral disease that you get all the time. Food poisoning and common cold are common examples. Now, I'm going to focus on bacterial diseases here because that's my area of expertise. But many of the more general points that I'm going to make about the evolution of infectious diseases, the way that the microorganisms interact with the human cells to cause disease, and then the way that we treat those diseases, a lot of the general statements actually apply to both, although the examples I'm going to give are all going to be bacterial. So our question is, how are the pathogens, that is the microorganisms that cause disease, different from all other microorganisms? And over the past couple of decades, there's been a tremendous amount of work done at the molecular level to say what are the specific genes or gene products that cause one bacterium to be pathogenic and another not to be. And I'm going to give you a couple of examples here that are all pathogens that work in the gut, that is that you eat or drink and then cause disease in the gastrointestinal system, that are all bacteria that my group has worked on. So we'll start off with cholera, which is caused by the organism Vibrio cholerae. Then a particularly severe form of food poisoning that's called by Listeria monocytogenes. And then epidemic dysentery that I just mentioned caused by Shigella flexneri. And I'll show you a little bit about why these bacteria can make you profoundly ill, even in numbers that are very small compared to the bacteria that normally live in your gut. So this is just another view of the gut. Here's the outside of it. If you open it up, you can see with the naked eye, you can see what are called these circular valves that are big folds in that tissue. And then on the surface of each of those things, there are these villi, these little fingers that stick up. And those villi are all covered with the epithelial cells that absorb the nutrients from the gut. And if you look very closely at just one of these little epithelial cells, it's got this really interesting shape where its nucleus sits down at the bottom, and then up on top it's got what are called microvilli that are little folds in the membrane that allow the apical surface area of the cell to be very large. So if you want to absorb nutrients from the lumen in the gut, you want to have lots and lots of surface area to do the absorption because that's the only place where your body can get food. So you get that extra surface area at all levels, these folds, these fingers, and then these tiny, tiny folds on the surfaces of the cell. So another thing that's special about these cells, the epithelial cells in particularly the small intestine, is that there's only one layer of cells that's separating the inside from the outside. That's also true in your lungs, again, where there's only one thin layer of epithelial cells that's separating the inside from the outside. And in both of those cases, it's because you need to be able to absorb stuff. You need to absorb oxygen in your lungs. You need to be able to absorb nutrients from your food. The rest of your body is covered with big, thick piles of lots and lots of cells. Like your skin is really thick and is really strong and really good and is a very good barrier between inside and outside. But the small intestine and the lung are very delicate. Those are your weakest defenses because those are the places where you have to absorb things. So as a general rule, this is certainly an oversimplification, but as a general rule, pathogens that cause systemic disease in healthy people will tend to go in either through the lung or through the intestine because those are the weakest parts of the defenses. So if we look at the intestine and we think about the intestine of somebody who has drunk Vibrio cholera in their water, what the bacteria do in there is, they'll get in there, they'll replicate. They don't have to be present in very large numbers, but they cause an effect on the host cell that then causes a profound change in the human's physiology. And specifically what they do is they alter the flow of salt and water through the gut cells. So normally these intestinal epithelial cells are taking up sugar, they're taking up amino acids, and they're also taking up salt and taking up water. What the cholera do is when they encounter these cells, they attach to the surfaces that have these little microvilli on them, and they express a protein called cholera toxin. That cholera toxin is secreted from the bacteria, and it attaches itself to the membrane of the host cell. And then through a series of steps, causes alterations in the physiology of the host cell, so that instead of absorbing salt and water from the gut, they now secrete salt and water through the gut. So this means you have massive watery diarrhea when you have cholera because the water, instead of going in, is now all coming out. And once it gets out into the gut, there's only one place that it can come out. Well, there's two, actually, both ends, and that's what usually happens with cholera. So the fundamental effect of this protein expressed by cholera is to cause dehydration of the cells, to cause the water to come out. And this is something we can look at in the lab at the cellular level by doing an experiment where we take human cells and we grow them on a cover slip so we can look at it on the microscope. And that's what we're seeing here, where this is an individual cell, this is a cell, this is a cell, this is a cell. You can see all these human cells are growing very happily all next to each other. They're making nice adherent structures to their neighbors. They like to grow in a big clump, just like they do in the epithelium. And this image is a phase contrast image through a culture of cells that are actually alive. We can keep them alive on the microscope stage by keeping them warm and giving the appropriate kinds of nutrients and the appropriate kinds of gases. And then we can do horrible things to them. So we can give them cholera while they're sitting there on the microscope stage so we can see what happens to all of the individual cells. So when I play the movie, as soon as the movie starts, there's going to be bacteria added to the culture. So there are the bacteria, the little black spots. You can see they stick to the surface of the cells. And now, as soon as they start sticking, they're secreting this toxin. And now what I hope you can begin to see is the cells are all shrinking and pulling away from each other. That's because they're being profoundly dehydrated. All of their water is coming out. We're now looping back through this and playing it again. So when a person has cholera, this is happening to their intestinal epithelial cells. They're losing water out into the gut. What that means is there's now more water in the blood than in the cells, so the cells take the water from the blood. The blood is dehydrated, and now the blood tries to take the water from all the rest of the cells in the body. So although the toxin is only touching the intestinal epithelial cells, all of the cells in the body are getting dehydrated. So a person with late-stage cholera, if you pinch their skin, for all of us, our skin bounces back very nicely. In severe cholera, you can pinch the skin, it'll stay because there's not enough water in there to make everything bounce back. So for adults, usually you can survive cholera if you just are given adequate hydration. But it's a very severe problem, either for people who can't get adequate hydration in medical care or for small children or old people. And this is why, this is what's happening at the cellular level. Now, what's going on with the toxin? So that's been studied at a lot of different levels. This is a case where you know what the gene is, we know what its function is, and we know even the structure of the protein. And it's this quite lovely structure shown here. It's a ring made up of several subunits that associates with the surface of the intestinal cell, and it's what's called a bipartite toxin, where this ring actually serves to open up a little window into the cell and then deliver the A subunit into the inside of the cell where it's able to have those effects. So this is an interesting case that I think is kind of really a general truth. which is that the symptoms of disease that are caused by bacterial infections in humans are not caused by the presence of the bacteria. They're caused by the fact that the bacteria, these pathogenic bacteria, send specific signals to our cells that change the behavior of our cells, and those are the symptoms we experience. Question?
What's the ultimate benefit to the bacteria if it ends up killing its host, thereby committing suicide?
So the question was, what's the benefit to the bacterium of doing this if it ends up killing the host? So in the case of cholera, there's a very obvious benefit, which is that when the cholera is actually in the gut, that's again a very warm, nutrient-rich environment, and the replication rate of cholera inside a nice, warm, nutrient-rich human is much faster than the replication rate out in the river. So if you think about the life cycle of cholera, most of the time it lives in the river. If it gets into a person, it will replicate like mad. You'll get lots and lots of offspring. And then that person has voluminous watery diarrhea, and all that cholera comes right back out and goes right back into the river. So it's actually, for that particular example, it's just a way to replicate faster. See why this is so good for an after-dinner talk? Okay, so the cholera toxin is a case where we understand it very well at a lot of different levels. And it's interesting, you know, actually the more we get to know the mechanisms of pathogenesis for a lot of different bacteria, the more we get to realize that even though the diseases may seem very different at the level of the human who's suffering from them, there's a lot of similarity at the molecular level in terms of what's going on. So for example, cholera causes diarrhea. There's another bacterium, anthrax, that has a toxin that is very, very similar. It has the same overall structure, you know, the same sort of ring structure where the ring then delivers a toxin into the host cell. But instead of going in through the intestine, anthrax goes in through the lungs. And so instead of causing water to come out in the intestine and cause diarrhea, the anthrax toxin makes water come out into the lungs and causes pulmonary edema. But again, the toxin, the molecular mechanism is similar. Now, there's some cases where we can have diseases that cause, you know, maybe fairly similar outcomes that have different molecular mechanisms, and this is also interesting. And, you know, one of the most diverse sets of diseases are the diarrheal diseases. There's many, many different ways that you can trigger intestinal cells to have some alteration in terms of their behavior that will give you diarrhea. So the other examples that I want to talk about are two bacteria that are also food and waterborne, Listeria monocytogenes and Shigella flexneri, that also interact with intestinal epithelial cells, but do so in a completely different way from how cholera interacts with them. So the cholera, remember, the bacteria stay in the lumen of the intestine, and they just deliver the protein to the cell, and then they get washed out when the diarrhea comes. Now, what Listeria and Shigella do is different, and arguably it's a lot sneakier, which is what they're actually trying to do is get to a part of the body that is sterile, that doesn't have competition from other bacteria. So they have to actually get in through that line of defense of the epithelial cells. And the way that these particular bacteria do it is they'll bind to the surface of the epithelial cell, just like we saw cholera do in the movie. But then they send a signal to the epithelial cell that causes the epithelial cell to actively embrace the bacterium and pull it inside of itself. That's what's illustrated here in step 1, where we go from the bacteria binding on the surface to coming into this membrane-bound compartment. And then what these two bacteria will do is secrete enzymes that break down that membrane, so now they're free right in the cytoplasm of the host cell. And once they're in the cytoplasm of the host cell, they start to replicate. There's lots of nutrients in there, lots of sugar. They love to eat glucose 6-phosphate, which is highly abundant inside the intestinal epithelial cells. And then they actually start to move around. And it's hard to move inside a cell because the cell is very dense. It's filled with stuff. So if you just tried to move like a regular bacterium by spinning a flagellum, it wouldn't work. So the way these bacteria move around is they take advantage of proteins in the cell called actin, which are part of the host cell cytoskeleton that I'll introduce in a moment. But they cause the rearrangement of these host cell proteins to make these very spectacular-looking structures called comet tails that actually push the bacteria around inside of the cell and then push them into the neighboring cell. Remember, in the intestinal epithelium, all these cells are sitting right next to each other like little palisades. But these bacteria can then push into the neighboring cell. Then they can do the same thing, break out again, and start replicating in the second cell. So in this way, the bacteria have gotten out of the lumen, they've gotten into the intestinal cells, and they can replicate there to their heart's content, where they have no competition from other bacteria and where they are invisible to antibodies in the blood. So how do they do this? Well, as I said, they take advantage of structural components in the host cell, specifically the cytoskeleton. And the part of the cytoskeleton they take advantage of are structures called actin filaments. So the cytoskeleton in general is a term for the structural proteins in a cell that cause the cell to have its shape and its ability to move around. And one of these proteins is actin. Some others you might have heard of are microtubules and intermediate filaments. These are all proteins where their primary function is to self-assemble to make structures that are much larger than individual proteins. And for actin, the way that it self-assembles is the individual actin protein is just one of these little red dots, but they like to bind to other copies of themselves in such a way that you can make long filaments that's just the same protein copied over and over and over again. And the place where this has been best studied is in skeletal muscle, where the contractile units that enable the muscle to shorten are made up of thin filaments that are actin and then thick filaments that are another protein called myosin, where the thin filaments and the thick filaments slide relative to one another to make the muscle contract. But you have the same kind of structural protein in other cells. For example, this micrograph shows those microvilli that are up on the surface of the intestinal epithelial cells that increase the surface area for absorptive purposes. And those also have these bundles of actin filaments in them. That's actually how that shape is maintained, how all those little invaginations are maintained, is by structural support from these filamentous proteins. You can also find them in lots of other cells making other kinds of structures. Now, in a cell that's been infected with Listeria, then, if you look at where the actin is, the actin here is labeled green and the bacteria labeled red. And what I hope you can see is every little red dot here has a big green streak coming off of it. But at the same time, there's plenty of other green streaks that are not associated with the bacteria. These parts here are the cytoskeleton the cell wants to have that help maintain its shape. These things here are called stress fibers. They help maintain the integrity of the cell. But the bacteria recruit this structural protein to make these little comet tails. And I'll show you what this does for the bacteria. This now is a time-lapse video, similar to what I showed you for cholera. But now all these bacteria are actually inside of the cell instead of on the outside. And this particular cell was infected about 5 hours before this movie was taken, and was infected with only one bacterium. So all of these little guys in here that are motorboating around, those are all the clonal descendants of that bacterium, which has been madly replicating over 5 hours. And you can see almost all of them are associated with these dark streaks, which is this host cell protein, the actin protein, that they've recruited to make these things, these units that will push them around. So my group has studied the mechanism of this for Listeria monocytogenes and for Shigella flexneri, but there's actually a whole bunch of different bacteria that do the same trick. And there's even one virus, vaccinia virus, which is the smallpox vaccine, which also does the same trick. And interestingly enough, in all the cases where we or other groups have tracked down the identity of the virulence factors, that is, those specific pathogenesis-related proteins that enable the bacteria to do this trick, it's always different. So these half dozen different bacteria that do it, they each have invented this mechanism independently of one another. So somehow it's something that's easy to do. It's obviously very good for the bacteria as a mechanism to spread around and spread from cell to cell. But it seems to be something that in evolutionary terms is really pretty easy to pick up. And you don't even have to be a bacterium to pick it up. You can even figure out how to do this if you're just a virus. So my group has looked a lot at the mechanism of how this works. And I just want to spend a couple minutes sort of giving you a flavor for how well we understand it. The bottom line is that the bacteria have a particular protein that they express bound to their surface that alters the assembly properties of this host cell protein, this host cell actin. In this image here, we're looking at a thin section micrograph where here's the bacterium. It's living in the cytoplasm of the cell. Here's a membrane-bound compartment in the cytoplasm. And this is its comet tail that's made up of all these little actin filaments. Now, each one of these little actin filaments is made up of about 300 copies of the actin protein that are all assembled in this head-to-tail fashion. And what the bacterium is doing is it's expressing this protein on the surface that induces the assembly of those filaments right immediately at the surface. And the assembly of this material pushes the bacterium. It leaves those filaments behind. It doesn't need them anymore. So this whole tail back here is sort of a history of where the bacterium has been. It's like the wake behind a boat. And you'll notice over here the filaments are a lot less dense than they are here. That's because over time the filaments disassemble. They fall back apart into those constituent subunits, those individual proteins. Those individual proteins then can diffuse around and they can assemble somewhere else. For example, this one disassembling over here, those proteins can diffuse around. If they find themselves back here, they can assemble again, and the whole thing can go in perpetual motion. And the bacterium is very lazy about this. All it does is express one protein on its surface, And in fact, once that protein is out there, you can kill that bacterium with an antibiotic. It'll keep jetting around forever. Because all it had to do was make it once, and then the host cell does all of the work. Yep?
How do you give direction?
How do you give direction? I could give you half a dozen publications from my lab about that. It's a very difficult question. The short version is the bacterium has figured out very clever ways to express that protein only on one pole. So it's basically got jet engines only at the back. But how it gets it just to that one pole, that's something that it took us about 8 years to figure out. Yep?
So the bacterium is using no energy.
That's
so
right.
It's stealing energy from the cell.
It's stealing energy from the host cell. That's exactly right. Yep. The bacterial protein is called Act-A. In Shigella, there's an equivalent protein. As I said, it's unrelated. The Shigella protein is called ICSA or VIR-G. And for all the other bacteria, there's different proteins, but they all do more or less the same trick. Yep.
So you can kill these bacteria with antibiotics, but
yes.
Have they, and the antibiotic also kills other bacteria, good or bad, at the same time?
Yes.
What work is being done to get more targeted antibiotics that kill less bacteria?
Yeah, so that's a terrific question. The question was, given that you can kill bacteria like this with antibiotics, but the antibiotics also kill off all the good bacteria, particularly in the gut, are there approaches where you could take to target specifically the pathogenic bacteria rather than the good bacteria? So that was going to be the second half of the third part of my talk. So we're not going to get to it tonight, but the short version is that yes, people are working on that. The difficulty is really an economic one. That there's 100 antibiotics on the market. Most of them work pretty well. There's resistance coming up, as you know, but resistance is not universal. Most of those antibiotics are reasonably broad spectrum, meaning they'll kill more than one different kind of pathogen. Now imagine you're a pharmaceutical company and you're trying to develop a drug that will only kill one particular bacterium. So first off, that means the physician has to know, this child who comes in with diarrhea has diarrhea that is caused by salmonella and is not caused by some other bacterium. Okay, so that's a diagnostic which often is not immediately available to the physician. Next, they have to, for some reason decide they'd rather use this, very specialized targeted antibiotic that is less proven compared to a standard antibiotic where there's a lot of clinical data about how well that it works. And, and third, when you develop a new drug, antibiotics for the most part are pretty cheap, compared to, not all antibiotics, but most antibiotics are pretty cheap compared to drugs that are developed by biotechnology companies. So you're aiming for a small part of the market under a difficult diagnostic circumstance where you want somebody to buy an expensive drug instead of a cheap drug. So the bottom line is that basic scientists are working on mechanisms because we're interested in understanding how disease works. But there is no company that is trying to develop a drug that will target Act-A. There's no market for it. Okay, was there another question?
So was that food poisoning, or is that how food poisoning works? Because we seem to deal pretty effectively with food poisoning in about 24 hours without antibiotics.
Yeah, so there's again, dozens of different organisms that cause food poisoning. This particular one, if you get it as a healthy adult, you might not even notice. You'll have diarrhea for a little while, but it'll go away. The places where it becomes very problematic are if you're a pregnant woman or if you're immunocompromised. And you can be immunocompromised either because of an organ transplant or because of HIV or because you're elderly. Now, because these bacteria will just spread around inside of cells and spread from one cell to another, they can cross barriers that normal bacteria can't cross. So in a pregnant woman, Listeria will get across the placenta and will kill the fetus. Now, normally the placenta is a very good barrier between the mother's blood system and the fetus's blood system, but the bacteria spread from cell to cell to cell. They go right across the placenta. If you're immunocompromised, it crosses the blood-brain barrier and it causes meningitis. So it's unlikely that you'll get it. Certainly as a healthy adult, you're not going to get it. Even though you eat Listeria every day, it's on your lettuce and everything, it's not a problem. But your immune system will deal with it. But if you're pregnant or immunocompromised, you eat a large bolus, then the difficulty is that the consequences of the infection are very severe. Okay, I think I'm supposed to stop in 4 minutes, so I'm trying to decide what's the most interesting, the single most interesting thing that I have to show you. Yeah, yeah, you don't know how fast I can talk when I really get going. Okay, there's two things that I'm going to show you because there are 2 more movies, and it's always great to watch the movies. So this is just to show a sort of quantitative point, which is, these bacteria move around by piling this stuff up. And if you try to scale that up to a system where you have good physical intuition, like the similar shaped submarine, which just like the bacterium is a cylinder with a hemisphere on each end, also leaves behind a characteristic curved comet tail as it moves. Looking at the bacteria, how fast they move, if you were to scale up a bacterium to be the size of a submarine, it would go just as fast as a submarine. So even though this is a weird mechanism, I mean, one thing it means is if you were in a satellite and you were like making a movie of submarines cruising around on the surface of the ocean, the movie would look identical to the movie that I just showed you of the bacteria. So it's moving pretty fast. You know, submarines have great big engines. But here's the trick. The submarine is moving through water. The bacterium is moving through a cell. If you actually calculate the numbers and say how much work is the bacterium doing to move through the cell, it's like the submarine moving through reinforced concrete at the same speed that the submarine actually moves. So this is a really strong engine. So this is the second to last movie I want to show you. This is an experiment done by one of my graduate students where she was trying to look at what happens when the bacteria run into stuff in the cell. Because if the engine is really this strong, it should be able to push anything out of the way. So here we're looking at a movie where the bacteria are green, labeled with GFP. And then the red dots here, which you'll see better on the next movie, are mitochondria. That is the energy-producing units in the cell that are quite large organelles. And in fact, they're about the size of bacteria. They used to be bacteria back in the day. And if you zoom in on one of these things, you look at what happens when one of these green bacteria runs into the mitochondria. When it loops back around again, I want you to see the guy coming in the top. It just doesn't even slow down. So this is an extremely powerful engine. Yep.
So is part of the reason when you get sick that you feel tired because these guys are using so much energy?
Well, you know, if you actually calculate how much energy is the cell using for cytoskeletal assembly and disassembly compared to everything else, it's a pretty small fraction of it. So it's not that you're actually de-energizing the cell or depleting the amount of ATP that's present in a cell. For any normal cell under normal circumstances, the amount of ATP is always present in excess, and it's really a rare thing to run out of ATP. That's like, you know, when mussels go into rigor, but it's very unusual. I believe the reason you feel tired is because of immune system reactions, but not at the cellular level. Yep?
So how does a normal, healthy adult body deal with that?
Okay. So thank you for asking that question. This is the part of my talk I'm not going to get to, but I said antibodies won't see these bacteria because they're hiding inside the cell. But there's a second arm of your immune system, not the antibody-mediated arm, but what's called the cell-mediated arm, where there are specific immune system cells that recognize cells in your body. They're expressing antigens, usually from viruses, on their surface as a sort of a warning system. That cell-mediated arm of the immune system will wipe out a human cell if it's showing signs of being infected. So the way your healthy adult body deals with listeria is it treats it like a viral infection. You don't actually kill the bacteria directly, but you kill the human cells that have the bacteria inside of them. And you can always make more human cells, so that's not usually a big deal. Okay, so I'm not going to explain why we care how it works, because I'm going to jump ahead to the very last thing I want to show you, which has to do with how we fight back. So the pathogens have these special genes that do things that interact with the host cell, very small changes compared to non-pathogenic organisms. And these new genes come from rapid evolution, as we were discussing, and they also come from transfer of DNA that can come from other sources. And this is why, you know, sex is better among the bacteria, because they can change DNA with somebody who's not even their own species, you know, as many times as they want. They can take a little bit, not the whole thing. It's very efficient getting DNA around in the bacterial kingdom. So let's see. So this is all the stuff I'm sticking. This is what I want to show you. How do we fight back? So the bacteria in there, they're getting all this DNA, they're evolving really fast. They evolve much faster than humans can. So we have to have specific parts of our bodies that are able to respond quickly and able to evolve quickly that can deal with this threat. And that's what our immune system is for. It's to evolve quickly and respond quickly. And in terms of quick response, the very best example that you're ever going to see in your lives is the one I'm about to show you right now. What I'm going to show you here is a movie that was made by David Rogers at Vanderbilt University in the 1950s. And in the 1950s, there was no digital image capture technology. What he did was he took a movie camera, a regular old movie camera, and he hooked it up to a microscope with some optics. And then he did a very simple experiment where he took some blood, some human blood, fresh blood, probably his own blood, and smeared it on a microscope slide, and then squirted in a few little bacteria. And then he just turned the crank on the, well, I mean, there was electricity, but he just let the movie camera run to record what happened. And so what you're looking at here is this blood smear, where these are red blood cells, And this is a particular kind of white blood cell that's called a neutrophil that is extremely good at hunting bacteria. And this poor hapless little thing over here is one of the bacterium that he had, and this is probably Streptococcus pneumoniae. And remember, this is done before there was any kind of time-lapse technology. So the movie, having been digitized, there's some problems with time, but it is almost real time, what we're going to see here. Now, in my field of cell motility, this is considered to be the greatest movie ever made. It's like the Citizen Kane of our field. And I've probably seen this movie 1,000 times, and I can't tell you how happy I am to show it to you tonight. So here we go. Now notice there's another bacterium down here. It sort of sniffs at it, but then it decides, no, I gotta stick on the main target. Sneaky. Yeah. All right, so the bacteria can evolve quickly, but they can't do that. So that's where I'm going to stop. The other topics that I was planning to cover had to do with immune system function and then antibiotics and how antibiotics work and how we become resistant to antibiotics. So if any of you are interested in talking to me separately about those kinds of topics afterwards, I'd be happy to. I'd also be happy to answer any other kinds of questions that you have. But I'll leave you with the neutrophil because it's the hope of the future. Thank you.
Well, I have to say, having spent the last 30-some-odd years studying infectious disease, you captured it in about an hour and a half. So that was a fantastic presentation, and I think a great prelude to more topics. We're going to have you back, Julie, for sure. So thank you for being here. Next week, we're actually going to continue some of this cholera theme, believe it or not, and we're going to move from the microbe to the host and the environment that sort of sets up the reactions. So this is part of a continuing set of themes, and we'll keep evolving from there. So again, thank you, Julie.
For more, please visit us at stanford.edu.